Okay, still half minute to go. It's fine. Okay, so I think it's about the time. Welcome, everybody. Welcome to our 19th seminar. Today, the title is Cellular Blood Flow Modeling with SDPD, namely Smooth Dissipative Particle Dynamics. So the speaker will be Professor Ting Ye from Department of Computational Mathematics, Jilin University, China. And Dr. Ye is currently the professor there. He received a PhD in 2012 from Nanyang Technology University of Singapore and focused on the simulation of cell manipulation under electric force during the PhD period. After that, he moved to the neighboring university, National University of Singapore, as a postdoc fellow. And there he worked on the cell modeling with DPD method. In 2014, he joined Jilin University, one of the best universities in China, and focused from then on the SDPD and application on the cellular flow in complex vessels. So welcome, Professor Ye. Now the stage is yours. Please go ahead.
Thank you, Bianxin. Hello, everyone. Thanks Zheng Li and He Li for the invitation. It's my pleasure to give a talk in this CFSM seminar. I'm Ting Ye from Jilin University of China. The title of my talk is Cellular Blood Flow Simulations with Smooth Dissipative Particle Dynamics. And we know the blood is important but complex. It is composed of 55% plasma and 45% cells. The plasma mainly consists of water with 91.5% and a small bit of proteins and ions. The cells mainly include red blood cells, taking up 99.8% in volume, and contain few white blood cells and platelets. When we perform numerical simulation, the first question is which components should we be considered? And this depends on the simulation scale and the problem to be resolved. For example, the platelets should be included when simulating the thrombus formation, and the WBCs should be considered when simulating the margination of cells. When talking about the scale, the blood research had three main stages. After William Harvey found the blood circulation in 1616, micro scale, cellular scale, and this micro scale. At the macro scale, people want to know how the blood flows in vessel and what the flow velocity and blood viscosity are. Initially, the blood is assumed to be Newtonian, and hugging porcelain law is a general formula. Later, the blood was found to be non-Newtonian, and the viscosity is 60 cp at 0.1 second minus 1, but 5.5 cp at 200 second minus 1. The different type of non-Newtonian models have been developed, and here I show one of them, which depends on the cell rate of gamma dot. Clearly, the non-Newtonian behavior is attributed to the presence of the cells. And there are two start points. One is including the cell information in the viscosity formula. For example, please develop such a formula, including the hematocrit of IBCs. The other point is, can we separate the cell from blood to model the non-Newtonian behavior, which leads to the cellular scale of blood research. At this scale, one often investigates the cell behaviors to understand the mechanism of some diseases. There are two main models, mixture cellular and the immerse boundary model. The mixture treats the plasma and the cells as two kinds of fluids, and each of them governed by the Navier-Stokes equation. The interaction between them is described by the force Fi. The immersed boundary model treats as plasma and cytoplasma as a single fluid, and the cell membrane is treated as immersed boundary. The interaction between them is described by the force Fs. Compared with these two models, the mixture theory can simulate a huge number of cells, but cannot describe the cell behavior. On the contrary, the immersed boundary model is able to couple the cell behavior, but cannot simulate a huge number of cells due to the computational costs. At the micro scale, people want to know the molecular details of the diseases. For example, how are the tumor cells captured on the vessel wall? At this scale, a single cell analysis is of interest, where the cell deformation, aggregation, and adhesion are particular noteworthy for understanding the relevant diseases. Here is a summary of such a blood research history. Currently, there are 3 scales for modeling the blood. At the macro scale, the blood is often treated to be Newtonian or non-Newtonian. The Newtonian model is still satisfied even now for some special cases. For example, larger vessel wider than 0.5 millimeter in diameter, or high steroid larger than 100 seconds minus 1, such as artery. For the non-Newtonian model, the focus is how to develop the reasonable constitutive relations. At the cellular scale, there are 2 models, 2 fluids, and 1 fluid with bodies. At the micro scale, 3 cell behavior in particular was investigated, but it's quite challenging because of unclear biochemical mechanism, complex mass models, or even no accurate models. In our work, we focused on the cellular scale and want to describe the cell behavior. So we use the immersed boundary model with 1 fluid and bodies. These are governing equations. So for such a model, how to numerically solve it? There are a lot of methods used to simulate the cellular blood flow. They are broadly classified into 3 groups: mass-based, particle-based, and hybrid method. Here I show some examples. The mass-based method mainly include finite difference, finite volume, and finite element method, as shown in these 2 figures. This is to study the RBC distribution with finite volume method, and this is to study RBC aggregation with finite element method. Note that there are few studies use FDM. The reason is the finite difference method often uses structure method, which is quite difficult to discretize the complex vascular networks. The particle-based method mainly include the SPS, DPD, and SDPD, as shown in these 3 figures. The first is use SPS, and this is use DPD, and the last in our work use SDPD. The lattice-Boltzmann method is one of hybrid methods, which have been widely applied into the blood simulation recently, such as cell aggregation, cell deformation, and cell suppression. So which method is the best one? We evaluate this from 4 aspects. From the viewpoint of accuracy, the particle-based method is generally less accurate than mass-based method. And it's difficult to say which one is more accurate between mass-based method and LBM. From the viewpoint of computational cost, the mass-based method is the best, but LBM may be the worst. From parallelization, we know LBM is very easy to be parallelized, so it should be the best. From viewpoint of implementation, I think the particle-based method may be easy. So in all, I think the LBM maybe is a better choice for current simulation, because the accuracy should be the most important factor, and then simulation scale. And the least important factor is easy to implement. That's just my opinion. But in our work, we still use particle-based methods because we hope to perform the multi-scale modeling and simulation in future, such as from MD to SDPD and then to SPH. When we numerically simulate the cellular blood flow, what are the challenges faced? Fred gave an excellent review in annual review fluid mechanics. He pointed out simulating the detail of such distorted cells as they pack closely and flow in complex geometries is the principal challenge of the cellular simulation of blood. More specifically, there are three main challenges. The first challenge is fluid structure interaction. Fred said such simulation must couple discretization of large deformation elasticity of cells with the viscous flow mechanics of the cell expansion. And the second challenge is multi-scale cell modeling. Fred said the nonlinearity of even the simplest memory constituted models is far more complex than that of Navier-Stokes equations, but likewise generates a challenge range of scales to resolve. The last challenge is to save computational costs. Fred said the flow of multi-cell system can be chaotic, demanding long run times to convert statistics of interest. Similarly, the microvasculature and many engineered devices have fundamental complex geometry. So when we simulate the cellular blood flow, this challenge should be resolved. Now I show the math models. This is a continuous governing equations governed by Navier-Stokes equation with immense boundary method. Using the standard procedure of SPH, this continuous governing equation is converted into discrete governing equation. We know that any continuous function can be expressed into this integral form with a delta function. The kernel approximation is use a kernel function W to replace this delta function. The particle approximation is to use a summation to replace this integral. So it's quite simple. Any physical quantity can be expressed into such a summation and substitute them into the continuous governing equation to form this discrete governing equation. These are our SDPD IBM framework. The IBM idea is shown in the third and fourth equations. The third equation reflects the action of cells on the fluid. Here, F diff and F aggregation and F adhesion are cell force, and FB is fluid force. So the memory force is converted to the fluid force, reflects the action of the cell on the fluid. The fourth equation reflects the action of fluid on cell. Vi is fluid velocity. So we use this fluid velocity to update the cell memory. Xk is cell memory. So reflecting the action of fluid on the cell. Here I don't want to introduce, I don't introduce the derivation of SDPD framework, but focus on the difference between SDPD and DPD. We know that for the incompatible flow, the discretization of viscosity term is not important. Thus that FD and FR have no large difference with DPD. Fg actually is gravity force to drive the fluid flow. So the first difference is the conservative force, Fc. In DPD, the coefficient alpha should be set manually, but in SDPD, it is derived from this pressure. And the pressure is generally used this equation, P is equal to rho C squared. Here C is sound speed, the speed of sound speed. We substitute this equation into the conservative force. We have the form like this. Here Fij is a weighted function, similar to the weight function in DPD. So the problem is convert to how to set alpha in DPD, or how to set the sound speed in SDPD. Okay, in SDPD and SPH, the sound speed C is set based on this formula. and it is well known as the CFL condition. In general, this formula gives a very large C, requiring small type step. And thus, sometimes one will set it arbitrarily. In DPD, the alpha is set by the empirical formula or even arbitrary set. Actually, this empirical formula is not accurate enough. Here I show an example of simulating lead-driven cavity flow. It can be found the particle distribution is not uniform if we use this empirical formula. But if we set alpha arbitrarily at one side, the particle seems uniform. Okay, the second difference is the result from the membrane force, FB. In our work, we use immersive boundary method to treat the interaction between fluid and cell. The membrane particles are not treated as SDPD particles. They are water particles for fluid. However, in DPD, the membrane is often modeled as DPD particles. Actually, this is not difference between SDPD and DPD, because DPD can also use IBM, also use immersive boundary method. Here, the advantage of using immersive boundary method is no need to set the physical parameter, such as the mass, pressure, and viscosity. But the disadvantage is low accuracy when fluid particle resolution is low. As the immersive boundary method results in this difference. So a conclusion is drawn that there are no major difference between SDPD and DPD. For the conservative force, we just need to satisfy this equation. In DPD, alpha I is, we need to set alpha IG. But in SDPD, we also need to set the sound speed C. So it is same, no difference. The alpha is arbitrarily set, and the C is sometimes also set in the same arbitrary way. If we set by the CFL condition, the time step is very small, so that the computational costs are unfoundable. Here I show some simple test cases. The first is calculate the self-diffusion. We can find the SDPD results much closer to the theoretical, the solid line in SDPD results. The dashed line is DPD results. We can find the SDPD results are much closer to the theoretical results than DPD. This does not mean the DPD is not accurate, just because the viscosity of DPD is also an empirical formula and not accurate enough. Okay, the second test is equator flow. The SDPD, DPD, and the SPH show the similar results. And the SPH curves are smooth because of without random force. Okay, the third test is Porcellier flow. The same results are observed. This is the SPH results, and here we can find the DPD and SDPD results. The last test is leader-driven cavity flow. The first figure is velocity field, and the second is velocity profile, and compared with finite difference method and finite volume method. The last figure shows the density and temperature profile. This is density profile, and these are temperature profile. We can find the density is not uniform for SDPD, DPD, and SPH. Here, please note that the DPD in DPD method, the alpha is set at 1 standard. Okay, we now turn to the cell models. These models are almost the same as those used in your group, so I just go through them. The first model is cell deformation model. Discrete model is used with consider the stretching and bending deformation, as well as that the surface area and volume almost conserved. Okay, the second model is describes the cell aggregation by a Morse potential model. It behaves as weak attraction at far distance, but strong repulsion at near distance. The third model is to describe the cell adhesion by a stochastic binding model. It is a linear model with forming and disrupting probability to describe the bond association and disassociation respectively. That's all for the models. Okay, the SDPD-IBM model has two sides. Clearly, it is suitable to solve the problem with complex domains and fluid structure interaction. And also, it may be potential in multi-scale simulations. But there are two obvious disadvantages. One is boundary condition, and the other is computational cost. In our work, we involve two types of boundary, solid boundary and open boundary. The solid boundary condition is a common issue of CFD method, but it is more difficult for the particle-based method due to the accuracy deficiency. In SDPD or SPH, when a particle is close to the solid wall, the support domain is not complete, leading to the accuracy loss near this solid wall. The open boundary condition is a common issue of particle-based method. The open boundary it is required to guarantee particle move into domain continuously, and also to set down the particle move out of the domain. So most of our prism work often use periodic boundary conditions, which is only effective for the case having the same inlet and outlet. In order to treat the solid boundary condition, we use a two-layer model. One layer is repulsive particles that are exactly on the solid wall to exert a repulsive force Fw on the particles close to the wall. The other layer is ghost particles that are outside the wall to complete the support domain and further improving the accuracy. This model can well avoid the cell penetrating from the solid wall, such as at the bifurcated positions, where the cell stays for a long period. In order to treat the open boundary condition, we developed an inflow and outflow boundary condition. This work was mainly finished by my former PhD student, Guanxin Li. Two more domains are added at the inlet and outlet, and named the inflow domain and outflow domain. The inflow domain is a generation domain that makes a particle move into the main simulation domain continuously. The outflow domain is a deletion domain that is used to set down the particles moving out of the domain. The other disadvantage is computational cost. That is also a common issue of particle-based method. Generally, the particle-based method adopts Lagrange description, in which the particles always move with the fluid field, different with mass-based method. As a result, the simulation accuracy is often low due to the non-uniform particle distribution. In order to improve the accuracy, a simple way is to increase the particle number. So that leading to the high computational cost. To solve this issue, we use a low accuracy but fast numerical algorithm, velocity wallet algorithm, to solve the SDPD-IBM formulation. It is a predictor-corrector algorithm. To solve the we first update the memory particle and then update the fluid particle. Based on these two predictions, and then we predict the velocity fluid. After that, we calculate all type of force, that's including the memory force and the repulsive force, Fw. Using this force, we correct the velocity field. That's the velocity wallet algorithm. It's a quite simple algorithm. We also use MPI parallelization to accelerate the simulation. It is a master-slaver configuration. The master thread is responsible for dividing tasks to slaver thread. And the slaver thread divided into two groups. One is for cell to calculate the memory force. The other group is for particles to update the velocity and coordinate of particles. Once all the results updated by the slaver thread, they are set back the master thread for outputting. That's all for numerical method. Okay, next we also some validation studies. First, we simulate the behavior of a capsule. For example, the deformation of capsule in this rectangular, stenosed, and bifurcated tubes. In the rectangular case, this is a 3D view, and this is a horizontal cross section, and this is a vertical cross section. The solid line is our results. In the stenosed and bifurcated result, and this is our result, and this is also our results. We can find the capsule shapes are close to those in other works. Okay, after that, we move to validate of the cells. This slide shows the deformation of cell passing through a slit. The top row is our result. Some typical shapes are observed, such as this all shape and two tug shape and the body shape in this case. Okay, this is for a cell passing through a stenosed vessel and compare with the experiment, and this is DPT result, and this is our SDPT result. Okay, one more for cell deformation validation. In this case, 5 cells pass through the stenosed tube. The left is our result, the right is SPH result. All of previous cases are for validating the cell deformation model. They are in good agreement with other work. And next, we validate the cell aggregation model. We simulated 2 cells and 5 cells aggregation under a standard condition and compared with this numerical and experiment results. At weak aggregation, the outer surface is concave, but at strong aggregation, it becomes convex. For 5 cells, the same phenomena are observed, concave at a weak stress, but convex and a strong aggregation. And we can find the other numerical results, and this experiment results also have this phenomenon. And this is for cell adhesion. We consider the adhesion of a spherical cell to a plate. The right column is our result. 3 different models are observed. This is detaching model, this rolling model, and the foam model. In the detaching model, we can find the cell is detached from the bottom plate. In the rolling model, the cell rolls on it. And in the foam adhesion, the cell is firmly adhered on this plate without any movement. Okay, the last test is to validate the open boundary condition. We examine the cell distribution in T-junction vessel under the different flow ratios. This vessel is a parent vessel, and this is labeled as the first vessel, and this is labeled as second vessel. The flow ratio is defined as Q1 over QP, and the cell number is defined as N1 over NP. And we can find the ratio of cell number increases as the flow ratio is increased. And Q1 over to QP and Q1 over QP equal to 0.5. The number ratio is also about 0.5. This figure shows a quantitative comparison. And the flow ratio, this is flow ratio, and flow ratio increased from 0 to 1 point. And the RBC flux ratio is also increased from 0 to 1. And at Q1 over QP equal to 0.5, we can find the N1 over OP is also close to 0.5. They are in good agreement. We also compare our simulate with Hagutakis. The left is our results, and it is surprised that even even the cell order is also identical. For example, we can find the 5th, 7th, and 8th, 6th, and 4th cells have the similar position with the Hagutaki's results. Here we can find this 5th cell and this 7th cell, and the last is 4th cell is the same. Okay, after these validations, we saw 2 simulation studies. The first one is about the thrombus formation. We first examine the motion of a single platelet or a plate without adhesion. This is well-known pole vaulting motion of a platelet. This figure shows a 2D view, and this is so 3D view. This movie. Okay. This movie shows the pole vaulting motion. We can find the platelet flips itself doing a pole vaulting motion. The right finger shows the comparison of platelet orientation and compare with this model's results. Okay, this case without the platelet adhesion. And then we include the platelet adhesion to reproduce the 3 adhesion model of platelets by simulating a line of RBCs and some platelets in a curved vessel. Note that the platelet adhesion is often regarded as the first step of the thrombus formation. This is a detachment model, where the platelets are detached from this thrombus surface. And this is rolling model. The platelets are rolling on this surface. And the last is foam adhesion, where the platelets are firmly adhered on the surface without any movement. The second important step of thrombus formation is the platelet aggregation. We also saw this process. The left movie shows the platelet aggregation and roll on this surface. And this is a cluster of platelets. And the right movie shows the aggregation, but firmly adhered on the surface. Here is a cluster of platelets. Okay, after examining these platelet behaviors, we studied other which condition a vessel is blocked. The first movie is a comparison basis, where the vessel is blocked. We have already known that the platelet adhesion and aggregation play an important role. So we elevate such blockage by reducing the adhesive stress or increasing the aggregation stress. Based on these questions, we designed 4 simulation cases. This is a weak adhesion of platelets, and this is a strong aggregation between IBCs and platelets. And this is a softer red blood cells. And the last is for large blood velocity. We can find the most effective way is to increase the blood velocity and then reduce the adhesive stress. The other ways have no obvious effect. These results support the effect of antiplatelet drugs and blood activating drugs. And maintaining high rate blood flow is perhaps more effective. Okay, that's the first simulation study. The second simulation study is about the cell distribution in complex network. We first simulate a successive bifurcation, and so in this movie. To compare with empirical results, we make some cross-section from P1 to P12 and measure the flow rate and red blood cell flux. Compare with our results with empirical results given by Price. We can find all of them are very close, except the value at P9. In In these movies, we can find there are no cells passing through these narrow vessels. And this vessel is the operation is P9. So the red blood cell flux is there. Okay, then we construct several more complex networks to examine the cell temporal and spatial distribution, which is difficultly measured by experiment. It easily found that the hematocrit distribution is different in different vessels. This is a spatial heterogeneous distribution. Moreover, for a same vessel, the hematocrit is also different at different time. This is temporal heterogeneous distribution. This heterogeneity are mainly attributed to the presence of bifurcations. Okay, that's the second simulation study. Lastly, I will give a summary. We developed a SDPD IBM framework to examine the cell, to simulate the cellular blood flow. It has some advantages, especially for treating the complex domain and fluid structure interactions. And it also potential in multi-scale modeling and simulation. And it has clear physical meanings and specific parameter setting compared with DPD. But it also has some disadvantages, such as high computational cost and low accuracy. Okay, I think that's my talk. Thank you.
Thanks, Dr. Ye, Professor Ye, for your very interesting talk. It's very systematic from SDPD and DPD comparison to the development of cellular motion by the SDPD. So if there is any questions from the audience, please just unmute yourself and speak.
So can I ask a question?
Sure, Go ahead.
So Tim is very good work, and either from the model and also for the computation efficiency, improve the computation efficiency. So I have a few questions I want to ask. The first thing is about the model. So when you consider the cell-cell interaction, you mentioned that you consider there's you use most potential just generate the repulsion force and the attraction force. I'm wondering for the cell membrane interaction, how do you consider the friction? Because you know that the membrane actually at the micro scale, if you consider that the membrane, the surface have a lot of kind of surface layer, if they contact, or they should also have this kind of friction force. So how do you capture the friction force between the cells? The most potential definitely does not consider this term, right?
Yes, I think it does not consider the friction force. Actually, when we use most potential model, the friction force should be neglected. Yeah, that's my idea. If we want to consider the friction force, it should be used some lubrication theory, but we have not used that theory.
But if you consider lubrication theory, usually that kind of thing is developed for the solid and the rigid surface. For soft surface, I'm not sure how to introduce lubrication interaction.
Somebody publishes work about the red blood cells use lubrication theory. So I think it should be okay.
Okay. The second question is about the simulation part. So you consider that the blood flow through very complicated vessel network. do you think the inelasticity of the wall will play a role in the flow pattern?
No, the boundary is solid wall.
Yeah, I know you are using the solid wall. I'm wondering if you introduce some inelasticity, where the simulation pattern will change significantly, or maybe it's a minor effect.
I think it should be changed significantly because this is moving particle when we change the boundary.
Okay. The third question is about on the page 14. So you consider the viscosity diffusion and the simulation result compare with the theoretical prediction. So we know in DPD actually people derive the theoretical formula, and they use the ideal gas particle distribution. That means they set the G of R equal to 1 and get the formula. So when you do the comparison, do you use the G of R equal to 1, or you introduce the particle distribution to get the corrected formula?
You means in DPD?
Yeah, in DPD actually, if you consider the DPD, the theoretical prediction formula is based on the ideal gas particle distribution. So I suppose this may be the reason you have a discrepancy between the simulation result and the theoretical result, because the theoretical prediction does not include the particle distribution at all.
Yes, I see. Yes, I see. I also treat the gas ideal gas.
You what? Can you repeat your
ideal gas? I also when calculate theoretical calculate the self-diffusion, I also use the ideal gas.
Okay, so this may be the reason why your theoretical prediction is off from the simulation results, because you use ideal gas particle distribution. This is not true for the.
But for SDPD, I use the equation of state is P equal to rho c square. It's also for ideal gas, right? But I get the result. We can find the theoretical and numerical results quite close. So for SDPD and DPD, here I use the same simulation condition.
Okay, but the theoretical formula actually is come from different duration.
So I think it should be this formula. The viscosity formula is not accurate enough.
Yeah, I suppose that the reason is because the theoretical prediction is the formula is wrong. It's not because the simulation is wrong.
Yeah. It's not simulation one. Yeah, right.
Okay, thank you.
Okay, thank you.
Hi, Yeting. I have two questions. So first, I wish to initiate a discussion with you. For example, you have been doing DPD, both DPD and SDPD, and eventually you decided to use SDPD to simulate the plasma, and then you model the cellular thing with the mesh. So, and then what do you see for the future of DPD? Is that DPD so bad, so people should use SDPD, according to your opinion? Because you show quite a lot of comparison between the two. And what do you see the future, how it goes?
Okay, actually it's hard to say which one is better. I think SDPD is the same as DPD. We can find the only difference is conservative force. In DPD, we should set the alpha, but in SDPD, we need to set the sound speed C. If we use the CFL condition, the time step is very small. The computational cost is very high. So in my work, the sound speed is set arbitrary. That is the same as set alpha ij in DPD. So I think it's no different.
Okay.
That's the SDPD and
Okay.
DPD.
I see. I
For
see.
future work, I think the first thing is we should be improve the accuracy for both SDPD and DPD.
Right, as a CFT practitioner,
Yes.
right? Because we see it from the CFT point of view.
CFT method, the result looks very good. But if you
Okay.
use this particle-based method, we know it.
Right. So then I have a second question. Speaking of CFD, so SDPD is one extension to the classical method of SPH. And then SPH is a solver of Navier-Stokes equation. And we also have other classical solvers based on mesh. And we have this issue. I mean, I have been doing SPH, SDPD both. So the particle method in general, especially SPH, is notoriously, expensive. So what do you see the advantage using SDPD or SPH in general compared to, for example, finite volume or finite difference in this case? You can do finite difference coupled with the immersion boundary, right? Doing the same thing. And why and what make you to decide to use SDPD eventually? So what's the big thing you get, that you don't have it for the mesh method?
Okay, that's me. So one slide. Actually, for a large number of cell simulation, the SDPD is the same as SPH. And many previous work, many people use SPH to simulate blood cell and use the finite element method as a finite volume method. It is macro method. But in our work, we still use this mesoscale method. That's because our start point is always we want to perform some multi-scale simulation. For example, we want to simulate from some molecular, such as cell membrane, cell proteins of the cell membrane, and then go to this SDPD work, and then go to a large scale SPH work. So we use this particle-based method. Otherwise, we prefer to the mass-based method because it's more accurate.
I see. So you favor the multi-scale nature of this
yes,
framework. I see. It's a good point.
So here we do not use SPH, but use this mesoscale SDPD because we want to bridge MD to SDPD.
I see. All right, thank you. Anybody else who has a question, please.
Hi, Dr. Ye. This is Jiao Yan from University at Buffalo. So thank you very much for the very interesting talk. So I have two questions about your work. So the first one, in fact, is about the, if you can go to the slide 37, then we can discuss from there.
Please.
37. Yeah, next one. Yes. So here, yes, 37. Yeah, this one can also work. So here you show some like a simulation results. So I can see the red blood cell and I can also see some blue particles. So what are those blue particles for?
The blue particle are not shown in these figures. I just show the cell and plate cell. Particle is full filled in this tube.
Yeah, so the blue particle is what are the blue particle? The blue
particle. Okay. The blue particle is platelet. This is platelet.
Oh, so
the red is red blood cell.
Oh, I see. So the blue
is platelet. The platelet is detached from this surface, this yellow surface. And the platelet rolling on this yellow surface, and the platelet hand on this. Yeah.
So are those platelets just a kind of single particle, or it has
no? It's about 200 particles.
200 particle. Okay, I see. And my second question, in fact, is about the mechanism for your thrombosis disease. So you show some kind of parameter study about like a different flux and the different maybe RBC like adhesion. Yes, this one. Oh, maybe 39, page 39. Can you please go back to page 39? Dr. Ye, would you please go back to 39?
Okay, it's quite
Yeah.
low. Okay.
Yes, that's one. Okay, sorry. So here, so you do a lot of like a parameter study here. So I'm just wondering what is the main like impact factor to which lead to this thrombosis like disease, like do you so from your like a comparison, which factor do you think is the most important one which caused this problem?
Okay, the most important factor is blood viscosity. We can find if we increase the blood velocity twice, this blockage is released.
Okay.
Yeah.
Then can that conclusion?
Yeah. But if we decrease the adhesive 10 times, this blockage is also okay. But these tools have no effect.
Okay.
So the most important factor, I think, is the blood velocity.
The blood.
In practice, it is quite dangerous to increase the blood velocity.
Then is it possible that the doctor may take this message and do something for the patient and solve the thrombosis problem? I mean, any kind of
okay, that's a good idea.
Yeah, I suppose this result just suggests you do more exercise.
Okay.
Yeah. Yes. Okay. So let's do more exercise.
Yeah. Yeah, thank you very much. But very interesting talk, and thank you. Yeah.
Okay, thank you.
Hi, Tim. So I have a question, Xin, and that's Phil.
Hey.
Yeah. Hi, Tim. Yeah, I'm Teng Zhang. I'm an associate professor at Syracuse University, and I know Zheng and Xian Jiaoyan before. And so I think I have first a very interesting talk. And I have a slightly not very related question and about this vascular network. I think you show that you want to understand the blood flow in the real kind of biological condition. And recently some colleague asked me about the vascular network structure. So my question is that how do you get the vascular network structure?
Okay, we use a CAD software, and first we get the CT imaging, face CT picture, and then we get the vessel center line, and then use CAD software get the whole structure.
Do you have kind of the hierarchical, hierarchical structure like a different diameter of the vascular network?
Yes, the diameter is changed with the actual direction. So
this is a standard process, or like I'm new to this field. I'm not sure it's like.
Yes, I think it should be standard procedure because this for the complex networks. The Professor Bakchi give a procedure like this. So actually, we follow his work to construct the complex network.
Okay, yeah, thank you. And I think related to the discussion about the mass and the particle-based method, definitely I think I'm not an expert on this, but I think the mass method, you need to take care of the unused volume, right? For the particle method, you just only need to discretize your real geometry. For the mass, like lattice Boltzmann or finite volume, you need to discretize the whole domain. Right?
Yes, correct.
Okay, thank you. Yeah.
Okay, thank you.
Is there any more questions from the audience? Please just speak up. Okay. If not, let's thank Dr. Ye again. Okay, it's very nice talk. Thank you for the efforts. Okay,
thank you.
hopefully I meet you in person very soon. Okay.
Okay.
Thank
Thank
you
you,
again.
Dr. Yeah. Thank you. Bye, everyone.
Bye.
，Michael 建议，
这样，李真。